Good morning, dear students. This is grade 10 science, unit 14, plant reproduction. I am Venilaan from Hindu College, Colombo. First, we will discuss about the definition for reproduction. Define the term reproduction. Define the term reproduction. The life process that gives rise to a new generation from an existing generation. Existing generation is referred to as reproduction. What is the reproduction method of bacteria? Already you studied about bacteria, that is kingdom Monera, prokaryotic. They don't have proper nucleus. So they produce very quickly. So what is the reproduction method of bacteria? That is called binary fission. Binary fission. What is mean by asexual reproduction? What is mean by asexual reproduction? Reproduction that occurs by spores and vegetative parts from a matured organism is known as asexual reproduction. Next, mention the organism which reproduced by the production of spores, producing spores. Mention one organism which reproduced by producing spores. Here you see the mushrooms. They produce spores. This is matured mushroom. They produce spores. So spores spread by air, spread through the air. So the organism is known as mushroom. So this organism produce spores spread by air. So spores spread by wind. Next question, what are the differences between sexual and asexual reproduction? So the asexual reproduction contributes only one parent organism. At the same time, they give rise to offspring which are more or less like maternal organisms. And the sexual reproduction contributes two organisms which are referred to as maternal and paternal. So give rise to offspring with a mixed characteristics of parents, because you studied the meiosis, the one type of cell division. So the cell division gives us the differences in the chromosomes. What are the differences between sexual and asexual reproduction? This is another one. The gametes are produced by sexual reproduction. So gametes mean the plant produces pollen grains and ova. At the same time, the animals produce sperms and eggs. But in the asexual reproduction, no production of gametes, no gametes. At the same time, the meiosis does not occur. What are the differences? So further, we continue the same question. So here the asexual reproduction, this is a natural method. So new characteristics are not formed. So no variation. But in sexual reproduction, you see the next generation has the different characteristics. New characteristics can be seen in sexual reproduction. Then another different, a large number of offsprings. Large number of offsprings can be produced in short period of time. At the same time, they can be seen in primitive plants and animal also, primitive plants and animals. But the sexual reproduction, increase of number of offspring is slow, increasing the offspring is very slow. Can be seen in plants and animal also, evolutionary advanced animals. So what is mean by vegetative reproduction? The process of generation, new plants from underground or aerial parts of a mother plant is referred to as vegetative reproduction. The process of generating new plants from underground, or the aerial part of a mother plant is referred to as vegetative reproduction. Here you see one example. I think this one is uh, ginger, like that. So this one were best example for the natural vegetative reproduction. So what is mean by natural vegetative propagation? Natural vegetative propagation. Generation of new plants naturally from underground or aerial parts of a mother plant is known as natural vegetative propagation. So next one, give some plants. So the natural vegetative propagation by root. So here the root also produce offspring, young ones. So this one guava. The second one is the root produces the next generation. This is breadfruit. Give examples for natural vegetative propagation by root. Already we studied two examples. So this one is the curry leaf. So some other plants, you can see the natural vegetative propagation by leaves. So here I mentioned two examples. First one is the begonia. The second one is the bryophyllum. In singular akapana, in Tamil Sadakarichan. Bryophyllum. These plants produce new generation by leaves. Another example also, here you see, this is common in Sri Lanka, queen of the night. You can plant the leaves like this. You can produce new plants. Give examples for the natural vegetative propagation by suckers. So here the suckers, that is little difficult to understand. This is chrysanthemum. Chrysanthemum is the plant. And what is mean by suckers? The small plant that rise horizontally from basal part of the stem under the soil is known as the suckers. The best example, banana, already we studied chrysanthemum. So what is mean by suckers? Small plants that rise horizontally from the basal part of the plant. Give example for the natural vegetative propagation by suckers. We studied one example, banana. What is mean by runners? Plant stems that can along the surface of the soil, connecting to the stem to the soil by adventitious foot are the runners. So I'll give some example for natural vegetative propagation by runners. This is very common example, centella. Another example, strawberry. Also another example. So these are the examples for the runners. What is underground stem? And mention the functions. What is underground stem? And mention the functions. Stems of plants which grow under the soil are known as underground stems. Vegetative propagation, storage of food, and spending dormant period. In some adverse conditions, these plants found it dormant. So these are some functions of the underground stem. Then they can tolerate the climatic conditions also. So the functions are vegetative propagation, storage of food, and spending dormant period in adverse climatic conditions. Give examples for natural vegetative propagation, rhizome. The rhizome, the best example, ginger. Give example for natural propagation by comb. Here you know this plant. This is called elephant foot jam. Another one, the natural vegetative propagation by bulbs. So leeks, onion also. Here you see the plant, the roots are fibrous roots, monocot plants. So this example is known as bulb. So give examples for natural vegetative propagation. Stem tuber, colus potato. This is example for the stem tuber. What is mean by bulbous? Bulbous. The best example is the pineapple. Pineapple. So what is mean by bulbous? Special reproductive structures form a modification of vegetative bud or a flower bud is known as bulbous. What is artificial propagation of plants? What is mean by the artificial propagation plant? Here you see the human involvement is here by cutting the plant stem like that. The process of growing many plants from one plant by man-made methods is called artificial propagation of plant. Now explain the ground layering method. So layering, that is very advanced method to propagate artificially. So here explain the ground layering method. So here first a small cut is made on underground for selecting twig. Then the twig is bent and buried under the soil. After a few weeks, the twig will develop roots. Then the twig is separated from the mother plant. So here another one is aerial labor, layering. Here you see the method of aerial layering. So this is also one method is very helpful to propagate easily. So this method is used the twigs which are high and above the ground. This method is used for the twigs which are high above the ground. So here we use some compost also. So we will explain later. So here explain the aerial layering method. A ring of bark of twig is removed. A mixture of compost and coir dust is placed around the place and tied with a strip of polythene. After a few weeks, the twig will develop roots. Then the twig is separated from mother plant. Here you will see the result, the pomegranate, lemon. So these plants, after layering, here you will see the roots are formed here. Yeah, here the root. This is adventitious root. So this is a result we can plant in a new place. Advantages of layering. What are the advantages of layering? So ground layering, already we studied this layering. So advantage of layering, the plants that do not produce seeds can be propagated successfully. So what are the advantage of layering? Another one, the several number of plants can be produced. So what is stock? So here, this is advanced method. So here the preparing stock, you see here the preparation of stock. So what is mean by stock? So the root part of the plant, the rooted part of the plant is known as the stock. Here you can see clearly they prepare the stock. So what are the characteristics of the stock? What are the characteristics that should be shown by a stock? The characteristics of stock and scion also important. Here bearing a strong root system. That is very, very important. Bearing a strong root system, having a uniform growth, withstanding the environmental changes and the bad weather, bad climate, lack of water also. So here, what is scion? So the twig or the bud taken from another plant and grafted to a stock is known as scion. Here you see the scion, the twig or the bud taken from another plant and grafted to a stock is known as scion. What are the characteristics of the scion? What are the characteristics of the scion? That should be shown in the scion. Here you see some plants that grafted. Here some advantages also there. So there should be a variety of good characteristics. So example, tasty fruit like that should be free from pests or diseases, should be free from. The scion determines the fruit variety, for example, flavor, aroma, texture. So here you see some examples. So what is grafting? What is bud grafting? Bud grafting. Here you see some bud grafting plants. So selecting a plant bud as scion, and grafting it to stock is known as the bud grafting. Selecting a plant bud as the scion, and the grafting it to a stock is known as bud grafting. Why does grafting can be done only on a dicotyledonous plant, dicot plant, not in monocot? The dicot plants have the cambium tissue. That is very important point. Cambium tissue, which is meristematic tissue, and is present in the vascular bundle. You know, the vascular bundle has phloem and xylem. So vascular bundles of the plant, that tissue has regeneration capacity. That is very important point. Which tissue? The meristematic tissue, the cambium, regeneration capacity, because of which it can form new tissue at the time of grafting. So explain twig grafting. So already you have seen that video. In this method, a twig is plant. Twig of a plant is taken as scion. Here you can observe clearly. So select a twig of a fruit bearing a plant, it has more suitable, and select a twig which have no tender leaf or a flower. So cutting the twig without damaging the cut. So this is the dicot plant. You can see the cambium in between the xylem and the phloem. So here you can see the twig grafting again. The next step, fixing the twig to the stalk so that the cambium are contacted. Wrapping the place from bottom to top. Very important point, bottom to top, not the top to bottom. Wrapping the place from bottom to top using polythene strips. See, carefully observe wrapping the place of grafting from bottom to top using polythene. Bottom to top is very important point, not the top to bottom. Yeah, to prevent entering water, to prevent entering the rainwater. Okay, so this is very important point. And graft and remove the wrap when the twig is observed to be growing. Then what are the advantages of grafting and budding? Advantages of grafting and budding. Production of offspring with the characteristics of scion. Obtaining of disease-resistant plants with strong root system. Propagation of plants that do not produce seeds successfully. So some more advantages you see in this mango tree. So the very fast production, and you can grow in a small space also. Another one, the variety of fruits, different colors, different taste can be produced by grafting. Mention the disadvantage of grafting. So the disadvantages: having a short lifespan, not successful with every plant, reduce wood value of the trees, low seasonal production. What is clone in tissue culture? The tissue culture is the modern technique. The artificial propagation of plants. The new offspring, which are identical to the parental plant, can be produced by cultivating any vegetative tissue of plant in the culture medium under the controlled condition. Offsprings thus obtained are called clones. So what are the components In the tissue culture, nutrient media. The nutrient media we have to prepare artificially. So sucrose, mineral salts, vitamins, and plant growth substances are included in the culture media. So what kind of plant tissue is suitable for the tissue culture? So generally tissues are taken from the apical bud or lateral bud or root tips in this for these purposes because they have the meristematic tissue. Meristematic tissue. What is the important steps followed in tissue culture? So the tissue culture, here the nutritive media has the tissues. They're introducing the part of vegetative tissue obtained from the mother plant into the culture media. So the important one, the culture media, you see the colors now developed, then allowing new roots and the buds to develop from the tissue called the colors. The colors here in this diagram you can see clearly which is grown from vegetative tissue introduced. So the next step. So here you see the next step of tissue culture, separating the plants because they produce a lot of plants. So we have to separate the plants and placing them into test tubes or some other flowers. Then next step, some other steps also there. Here this plant is potato. Potato also can be produced in tissue culture method. So next step is gradually let the new plant to get adapted to a natural condition to be cultivated in the field. So some advantages also there. So first advantage, the new plantlet can be grown in the short amount of time. Only a small amount of initial plant tissue is required because we use very small tissue. So the small tissue is enough. The new plants are more likely to be free of virus or diseases. On a large scale, the tissue culture produce help to supply the consumer market with new subspecies and varieties. So we can produce a lot of plants here. You need only relatively small space to perform the process. So for example, 10 times the plant in 1/10 of the space can be obtained a large number of plants using hybrid tissue with a favorite gene. So name some plants which can be produced by tissue culture. Here you see the plants we produce by tissue culture. This example, pineapple. So the second one, banana also. You can see very small bananas. So potato also here. Potato also can be produced. The paddy also can produce by this method. So this one is very helpful, but a lot of advantages are there. The advantages, ability to propagating plant that do not produce seed successfully, and ability to having offsprings that are identical to mother plant, ability to having offsprings that are identical to mother plant, propagation of plants that bear fruit. So the next one, ability to propagating selected plants which are resistant to disease and pest. Ability to generating plant varieties which withstand harsh environment conditions. So some disadvantages also there. The tissue culture can be required more labor and the cost, money, the sophisticated equipment also. There is a chance that propagate plants will be less resilient to diseases due to the type of environment. So some sort of disadvantages also there. I think this one will help you. This one just a revision for you. You already studied in this lesson, but thank you very much. Give your comments in the box. Thank you very much. I am Venilaan from Hindu College. Thank you.